Great. Well, thank you for joining today. So our webinar today is called Achieving a Higher Imaging Throughput with Synchronized Autofocus and Motion Control, co-hosted by Dover Motion and WDI. So just before we get started, a little bit about both of our companies. So Dover Motion, we're focused on precision motion axes, X, Y, and Z, especially for microscopy applications. And we also can provide higher level motion subassemblies. And for our Smart Stage ecosystem, we've developed the Motion Synergy API. And what that's designed to do is allow customers to get started quickly with a user interface that gives them motion quickly, and then also use that same platform to code their motion programs into their instrument design. Kevin, why don't you explain WDI?
Perfect. So WDI is an OEM microscopy automation solution company. Autofocus has been the core of our business when 20 years ago we created the first laser autofocus sensor for the flat panel display industry. Since then, we've expanded it into other sectors like the semiconductor wafer inspection, but then particularly relevant to this webinar, we are in the life science and biomedical imaging applications as well.
Hello, I'm Dig Thomas. I'm Principal Systems Engineer at Dover Motion. My educational background, long ago, I did my undergraduate in biomedical and electrical engineering, my master's in electrical. After that, I spent 10 years in Silicon Valley working with various startups, mainly focused on carbon nanotubes for materials and display applications. Moving on to semi and instrumentation and metrology. I'm currently in my 9th year with Dover Motion, serving as systems engineer.
And I'm Kevin. I work for WDI. So I did a physics degree at the University of Waterloo. I then worked as sort of a software verification engineer for a couple of years. And then I've been at WDI as an application engineer for about 5 years now, working with our customers to find solutions for everybody.
Great, thank you. So during the webinar, feel free to ask questions in the chat, and we will be covering those questions at the end. So feel free to please enter those. So our agenda today, you can see here, basically we're going to go through some application examples for both sequential field, so moving, stopping, imaging, and how autofocus and motion can help you increase throughput for those applications. We'll be talking about well-to-well, slide imaging, flow cell imaging as well. And we'll also touch on scanning. So if you need to do imaging and focus adjustments on the fly while you're scanning, we'll cover those applications as well.
So the main topics here today are about life science applications. So for many life science applications, you guys require fast and accurate autofocus and motion in challenging conditions. So we're going to explain how together the PFA dot sensor from WDI and the Dover DOF5 stage, as well as their smart stage, which is XY, meet the demands of the challenging applications. And particularly, we're going to cover two main topics, which is the multi-well plates and flow cells, because that is typically what we see in most customers and most clients, potential clients want to deal with.
So in terms of different types of autofocusing, there's sort of the pre-map where you take time before you process your sample. And typically with a contrast vision-based system, you go through and map multiple points on a wafer or whatever your sample may be. This is fairly time-consuming, takes some data crunching. It's fine if your process takes 4 hours, like for a wafer or something, and you have 10 minutes at the beginning. It's not really a big throughput hit. But if you do need something that gives you higher throughput, the best choice is really to go with on-the-fly, real-time autofocusing, where as you're moving along the sample, you're following the surface and focusing with real-time autofocus.
So when we talk about different types of autofocus, there's really only two options here. So there's the video autofocus, which is a contrast-based autofocus, and a laser autofocus, which is what WDI mainly covers. So with video, there are some pros. It's really cheap. You can implement it with basically any algorithm that you can find online or yourself. And it's really repeatable on a pattern medium that sometimes, if it has too many surfaces, maybe the laser autofocus rather, would have trouble with all those multiple surfaces happening all at once. But it's extremely slow. It doesn't work with any sort of clear glass or objectives like that where there isn't a pattern to reflect off of. And you, again, if you need to scan at all, this is not going to work for you. So the benefits of having a laser autofocus is it's super fast. You can do on the fly, as Dig was saying. It's accurate, precise. We advertise about a quarter depth of focus is the accuracy of our autofocus unit. Some cons is that it does require a refractive index change for the most part to identify a surface that you're going to focus on. And it requires more optical integration, so you need to sort of integrate it into your microscope in a way that makes sense. So these are two products that we're sort of talking about today. On the right side, you'll see our PFA dot sensor. That's what you will be integrating onto the microscope, into the infinity corrected microscope. And then on the left here, you see Dover Motion's DOF5 stage, which is a single objective stage that we send signals to command how to move to focus. So the principle of operation for our sensor is it's a through the lens triangulation to determine the distance and direction to focus. You can see on the right-hand side, the image that you can see there. When we're in focus, the laser focuses down to a nice small little dot. And then when we're above and below focus, it expands out to this sort of semicircular shape. And then depending on how far away we are, we can determine the distance and direction to focus, and then command the Dover stage such that you come back into focus. So these are the sort of mock-ups of the samples that we might be interested in. So these are some sort of cross-section of a flow cell and then sort of a cross-section of a typical multi-well plate. How challenging each of these applications is to have reliable autofocus is influenced by a couple things, like I've listed here. So the numerical aperture of your objective lens has a heavy influence on how we identify surfaces, and whether or not we detect the wrong surface by accident or not. How large the refractive index change is between those surfaces can have an effect. And then when you're scanning into multiple planes of interest, as you can see on the right side there with the multi-well plate, I've sort of drawn these two red lines within each well, and that sort of represents floating medium or floating biological bacteria or biological samples that you might want to image, and they aren't directly on that glass water refractive index change surface. And so depending on how deep into the well you need to go and the numerical aperture of the lens, etc., that can pose a challenge. So in this slide, I'm going to explain what we're looking at here. This is a screen from our autofocus console. So we have a software console that is mainly used for initial setup of the device in terms of aligning the system, but also to determine what sort of focus and what sort of sample we're looking at. So in this particular example, this is not a biological sample, but the surfaces are nice and far apart, so it's easy for me to teach people how to read this graph. So in this particular example, I have three surfaces that the autofocus sensor identifies, the PFA dot. We have the first, second, and third surface, and those surfaces are identified by when the yellow line here crosses the x-axis. Of particular interest here that I want to note is where I've identified the second surface capture range, and that's sort of between about -300 microns and +800 microns. And so what I mean by that is that if you were to have the DOF5 stage at, say, 600 microns, and you were to activate the autofocus, you would come back to that second surface of interest. And that's your, let's assume that your second surface is the one that you want to focus on. Outside of that range, so let's say you're up at 1,000 microns or a millimeter away from your focus position. If you were to select autofocus there, then you would travel to the third surface. So it's these sorts of problems that we try to solve through software and identify what surfaces we want to see and what surfaces we are seeing. So moving on to this slide, this is of a flow cell. So on the top, that where I've identified the top surface, that would be your first glass air interface, if you're imaging from the top. The first surface I've identified is sort of that first glass water interface, and then the second surface being that second glass water interface, so inside the channel of that flow cell. So you can see how in this application, the distances between that first and second surface are a lot smaller than we saw in the example that I had before, just to illustrate the point. So it's these types of challenges that we like to solve, and we'll be identifying that a little bit later. So these are the types of things that we use in software to help someone detect and stay on the correct surface. So the first thing that we might try is lowering the laser power. So what this does is it minimizes the signal from the undesired surfaces. Now you might be thinking that it also minimizes the signal from the desired surface, and that is true. But once we've locked onto that desired surface, having a lower laser power doesn't matter as much, and it helps you stay on the right surface. Another thing we can do is set motion limits. So the nice thing, especially with Dover, with the DOF5 stage, is because it's an encoder-based system, we can set really soft limits on the DOF5 stage such that it can't be moved past a specific encoder position. So if your surface is at, let's say, plus 100 microns on the encoder, and your surface that you don't want to go to is, let's say, 300 microns away, then you might set the motion limits to about 150, such that you can't move past and start detecting that wrong surface. Something else we can do is change the linear range, which is basically just minimizing the area of the sensor that we look at internally in our autofocus sensor. And that can help such that we don't even see a surface. And you're going to sort of see that later. In another slide, I do something else, and I can show you how that might relate to that. The fourth thing here, which is AY freeze range percentage, this is the only one that applies only to the scanning applications. All the other ones apply to both scanning and stop and go applications. So with the freeze range percentage, I would think of it more as like a moving average of the motion limit. So if it starts detecting some signal that wants to send the autofocus off to a far distance, we're saying, you know, just ignore that part and just stay locked on the place you are right now. So once we've sort of exhausted software ways of helping you stay on the correct surface, this is where we might change and start focusing on more hardware or physical mechanical things to do. So the first thing we'll talk about here is purposely misaligning the sensor. So what this is going to do is, at the sacrifice of a little bit of capture range, which I talk about in the next slide, you can separate surfaces. So in this image right here, you see that nice peak in the center that's nice and thin. So that's the surface that we want to focus on. So we're on that surface right now. And then on the left side, you see this big peak off to the side, and that's the other surface interfering with our signal. You can go to the next slide now. So here I see, by purposely misaligning the sensor in physical space, I can separate the surfaces such that when I'm focused on either of them, either on the top glass surface or the water glass interface, the sensor no longer sees that surface, and it has much of a less of an effect on the overall focus. So the second hardware thing that we can do is called C-Sync. So this is an optional digital input, which temporarily disables the laser and the autofocus when it is asserted. So this is useful for a couple of reasons. So let's say that you've mapped your X and Y, using the Dover Smart Stage, and then you want to turn off the laser when you're not inside of a well, when you're imaging, let's say, a multi-well plate. So this would avoid the PFA dot targeting the bottom, like glass air surface, when you're moving between wells or if you're moving outside of the well, of the multi-well plate itself. Furthermore, with like a flow cell, same thing. Let's say you have it all mapped. You know exactly where your channels are and where the edge of the flow cell is. So you want to avoid accidentally targeting the top surface, not the glass water interface, when moving between those channels or moving off the flow cell to, let's say, another flow cell in your process.
So sometimes you may not necessarily want to follow a single surface. You may be trying to image something three-dimensionally, go through and take a Z-stack of images. So direct drive is the way to go with this. It's high speed, high resolution. Another great advantage of direct drive is you get a nice travel range. You're not limited to a few hundred microns. There really is no limitation. So what we have here is the Dover's Smart Stage Z50 with counterbalance. And shown on the left is basically a typical illumination integration period for the image, our stage steps, there's download and repeat. But as you can see, the Z-stage move time is almost insignificant compared to everything else. And looking at the graphs on the right, you can see this high-speed performance. These 250 nanometer moves basically take less than a millisecond and settle within plus or minus 40 nanometers, which is phenomenal, again, if you're trying to do that three-dimensional imaging or any kind of characterization of more than a single surface.
So on this slide, we're talking directly just about multi-well plates. So we talked before about how maybe freeze range percentage or motion limits can help you. So in this case, you can see, between the wells, we don't have that any glass water interface. All it would see is the bottom of the plate. So by setting in some motion limits or maybe setting in the freeze range percentage, we can stop it from traveling back down to that bottom plate. Something I didn't mention before, but I touched on at the beginning, is how you might have multiple planes of interest within that multi-well plate or within any other biological sample that you have, where things aren't necessarily at the exact same plane, and the depth of focus of your objective is so small that the biological material needs to be imaged in multiple planes to have it happen. And I think Dig is going to talk about Z-stacking and that sort of stuff here. But if you were to want to scan across that same region of interest, then this is where we might introduce a different Make Zero location. So Make Zero is basically a training program where we say, I know that this is not the best focus that our laser thinks, but we're going to tell you that this is actually what we want to image. And that allows us to lock on a plane of interest that isn't necessarily at the refractive index plane. That being said, there are limitations on how far into the well you might be able to do that, and that's mainly dependent on the numerical aperture of the objective itself. But it's just one more tool that we use to get the results that our clients need.
So if you're doing well-to-well imaging sequentially, Dover has a technology called RME. It's repetitive motion enhancement. And again, you can see on the bottom left-hand side, similar to as before with the Z-stacking, you have this illumination integration period and download and move time. And we're to the point where our motion is so fast now, it's really on the order of the download time, or in some cases faster. So first plot in the top right shows a fairly well-tuned move settling in under 77 milliseconds. 2 millimeters to 200 nanometers. But then if you look in the bottom right, all of the resonances are gone, vanished. And this thing settles way, way faster. And this, again, is with our technology of RME, and it really allows you to get much higher throughput when you're doing that sequential field imaging. So conversely to the sequential field imaging, there's the scanning type imaging. This can be on a bowed or I guess it could be bowed or tilted sample. Long ago in industry I used to work in, we called them potato chips, and had various curve fits for them. So in any case, you need an autofocus to track that while you're scanning, and that can be imaging, that can be laser processing. Something you can do ahead of time, or you can do on the fly. But the second item that we have here for scanning imaging absolutely has to be done in real time. And this is something that really, only technology like WDI has can allow you to reach this level of performance where you actually perform autofocus and cancel out any sort of external vibrations that may be printing through in the Z direction while you're trying to focus. Again, this can't be a gigantic vibration, it can't be insanely high frequency, but there are many realistic signals that print through that through the use of WDI's autofocus and DOF5 or Smart Stage Z50, we're able to cancel out and get beautiful high resolution images. The last for scanning, so there's TDI, time delay and integration, where you have a camera that is synchronized with position trigger. And as that moves along, it integrates the signal. Benefits are less light, so less photo bleaching, things like that, and higher signal to noise ratio. Again, that's something you absolutely need a real-time autofocus tracking for.
So this is a capture taken with, again, the WDI's autofocus console. And this is part of our diagnostic suite to determine sort of how good our autofocus is. And this is sort of a report card on how well we think we're doing in terms of keeping up with your sample. So in this particular example, we're looking at a tilted or bowed sample. We find that this is mostly relevant to like a flow cell application where oftentimes you have it sort of clasped on either end and then maybe have a vacuum chuck in the middle that's sort of bowing the sample down. In this particular example, we have a 4 millirad tilt to it. And so that what you're looking at here with the green line is sort of the Z motion that we're going through over about the period's about 2.2 seconds. And then the magenta line is sort of our sensor readout of our position. And down in the corner, the bottom right-hand corner, you'll see min, max, standard deviation, and then RMS. So we're getting an RMS of about 10.4 in this case. And then we multiply that by a value for determining slope and the depth of focus. So in this case, anything up to 50 would actually be an acceptable value and keep your sample in focus. So we're doing very well here and well within the quarter depth of field that this objective needs to be maintain focus. And then this one is for the vibration issue. We injected a sort of 10 hertz, 600 nanometer peak-to-peak movement that our autofocus sensor had to track along with the DOF5 stage. In this case, we're getting an RMS value of 15.6, which again is well within the depth of focus of this particular objective. So it's really a testament to both WDI's PFA dot sensor as well as Dover's DOF5 stage that allows you to keep up with that sort of Z motion.
So continuing on with scanning and that trigger position theme, Dover has developed higher end encoder technology, which is called the ultra encoder, but it's really a very low subdivisional error encoder. And you can see in the bottom left-hand graph, if you squint, basically on the y-axis, you have cyclic error, which more or less dominates that subdivisional error. And on the x-axis, you have displacement. So in this case, it's 150 millimeter travel. And if you look at the plot, basically, few points it exceeds outside of plus or minus 5 nanometers. But our 3 sigma on that is 4.03 nanometers, which is phenomenal. And where this is really valuable is for where short range accuracy is important. So in sort of that hundreds of nanometers to a micron, where you can't tolerate too much variation in there, this knocks it out of the park. Obviously, it is very good at the long range accuracy as well, but this sort of covers both ends of the spectrum. And again, where there's value in this is for this trigger on position type technology, which again can be for cameras where you're sucking in photons, or lasers where you're firing them out. And this bottom right graph, we have sort of velocity on the top, and then 2 digital signals. The blue one is the gate, and the orange is would be your sort of incremental trigger on position signal. And this gate signal is useful for some customers to tie into either laser shutter, laser gate, illumination, and then they're able to acquire their images once they have their illumination on. And again, this is advantageous. You don't leave the light on all the time. You can be using laser illumination, and it can also help prevent any photo bleaching.
So there's really 2 options for interfacing between the PFA dot and the DOF5 Z stage. The first one I'll talk about, which I'm the most familiar with, is WDI's fully integrated solution. So this one's very simple. WDI software handles everything. So you would use our SDK to interface with Dover's Z stage. It's just one piece of software. And I'll be showing you in the next slide how it sort of looks to set up. And then you have this sort of hybrid solution. So that would be useful for way more demanding applications. Something like if you're doing really, really quick Z stacks, that might be where you would want to use Dover Motion's motion synergy software to help get the best performance out of the Dover DOF5. And that would require interfacing between both of our SDKs, Dover's and WDI's. And the sensor PFA dot would just send out step and direction signals to the DOF5 stage. So this is sort of what our system configuration looks like if you were to choose the first option, where we interface directly with the DOF5. You just select DOF5 encoder 5 nanometers in the Z motion section right there. And then over in the DOF5 panel right here, the DOF5 stage has hard stops. And so you can configure those hard stops any way you want, up to about 6 millimeters of travel total. And then we handle all the limit calibration for you, just you calibrate limits, and then that gets saved into the sensor. And you can home the stage and engage and disengage the stage from here.
So for the more advanced users, there's the hybrid solution. And this is for people who want complete control from a software perspective over the Dover Motion hardware and independently of the WDI hardware. In this type scenario, WDI sends a hardware signal, pulse and direction to the DOF5 or Smart Stage Z50, and that is basically the link between the two. All of the software outside is controlled by the customer via each individual software package, SDK. So for this pulse and direction, you can have a granularity of basically one count of the encoder, which is 5 nanometers. So if you have 200,000 steps per millimeter, that's your gear ratio of 1:1. If you want something a little coarser, never thought I'd say 25 nanometers was coarse, but you would do a 5:1 or 40,000 steps per millimeter. That would be set through Dover's Motion Synergy API. There's a little code snippet. Alternatively, you could set it in the Motion Synergy GUI as well.
Great. So just wrapping things up, some other available solutions. We touched a lot on our Smart Stage XY and DOF5 for Z focusing. We've got a whole suite of Smart Stage products where the controls and encoder are embedded in the stage in a small footprint. So currently, we're working on an open frame, which is in alpha. We've got a monolithic XY for better stiffness and lower profile applications. Our DMCM boards can run pretty much any linear motor or stepper motor stage that we manufacture. We've also got longer travel shuttle stages for applications that need maybe multiple shuttle axes on top, or just that longer travel for load unload.
And then WDI, the PFA dot is a laser dot based sensor. We also sell a laser line based sensor, either the ATF6 or our PFA sensor. It's exactly the same package. You can sort of see it there. It looks almost identical, and it still uses through the lens triangulation. But as you can see, instead of a dot, it sort of expands out into a rectangle. And this is good for highly patterned samples, because then we get multiple areas of feedback and can sort of discard extraneous signals that we maybe don't want from other artifacts within the same image.
So, so that wraps it up for today's presentation. Thank you for attending, everybody. Please enter questions in the chat. We do have a couple that came in already. First one was about the recording. So yes, after the event, the recording will be available through this link, and we will also send out an email to all attendees with the link for the recording. We've got another question that came in. So what is the smallest move the DOF5 can do? Dig, do you want to handle that one?
I believe we bet at like 15 nanometers. If you need something smaller than that, we could talk about it. It's certainly capable of doing smaller moves, but in terms of what we typically attempt, 15 nanometers.
Great. Any other questions? Okay, there's no more questions at this time. We'll wait another minute or two. Feel free to pop those questions in the chat. We've got another one here. So what would the advantage be going from the WDI ATF4 to the PFA dot?
Right. So we have a really high increase in response rate. So we're going from 1.2 kilohertz to 8 kilohertz. So in that case, we're getting a lot faster and more reliable signals back. So we can increase our throughput and increase the way that we analyze the signals.
Great. We'll wait a few more, another minute here and see if there's any follow-ups or additional questions. Okay, with that, it looks like no more questions coming in. So thank you, everybody, for attending, and look forward to future webinars.
All right, thanks, John.
Thank you very much.